Hello, welcome to this screencast where I'll be talking about blood types and a little bit about blood transfusions. So your goals here, by the end of this, you should be able to identify and describe what an antigen and an antibody are. Talk to me a little bit about the ABO blood groups or the ABO blood types, as well as the RH blood groups or blood types. So you should be able to then also explain agglutination, the basis for transfusion reactions and blood typing, as well as being able to then identify and explain who the universal donor is, who the universal recipient is. And then in class, we'll be able to apply this knowledge to describing and explaining the hemolytic disease of the newborn, or erythroblastosis fetalis, a pathology associated with blood typing, or the blood types anyway. So let's get started here. An antigen, as the title suggests up top here, antigens are cell surface proteins which cause the immune system to produce antibodies. So here we have in the center visual of this is the antigen, and the antigen has different shapes to it, here, here, and here. In this example, there's three shapes. Some antigens just have one shape, but these parts of the antigen have the different shapes, are known as epitopes. And that's kind of secondary information. But these antigens have a definite specific structure. The antibodies then here are these Y-shaped proteins. And these Y-shaped proteins have parts of them that will bind to the antigen. And that part that binds to the antigen is complementary to the antigen. Complementary to the antigen. So an antibody is specific for an antigen. And since they are Y-shaped, they can bind more than one antigen at a time. So again, an antigen, just by the way, is anything that will cause the immune system to produce antibodies or to generate antibodies. So this is where the antigen comes from. So this substance here, this antigen, is an antibody generator thing, right, that will cause the immune system to produce these antibodies. Okay, so in terms of blood vessels, in terms of blood cells here, this antigen and antibody stuff is important. So we have this table here, and we'll divide it into top and bottom. So the blood types are due to proteins or antigens on the surface of the red blood cells, right, or erythrocytes. So this is a red blood cell, and if you are blood type A, then you have on the surface you have A antigens, right, and which they're representing as this sphere. If you're blood type B, then your red blood cells have a B antigen on the surface. If you're blood type AB, then you have both A antigens and B antigens on your surface, so A antigens and B antigens. And if you are blood type O, then you do not have either A nor B. Right? You have neither A nor B. I sometimes also think of this as a zero, right? There are zero antigens on the cell surface. Okay. Now, as you know, blood is not only the cells, but is also plasma. So in the plasma, this is where you have proteins, and this is where you have antibodies. Right? Now, if you are blood type A, you have anti-B antibodies, and you notice that this antibody here is Y-shaped, right? And then this is the binding area right here. And so this antibody is specific for B antigen, not for A antigen. So in your blood, if you're blood type A, you have these anti-B antibodies. And notice that they will not bind to your red blood cells, because you don't have, on your red blood cells, you don't have the B antigens. If you're blood type B, you have anti-A antigens in your plasma. If you're blood type AB, then you have no antibodies in your plasma. And if you are blood type O, you have both anti-A antibodies and anti-B antibodies. Okay? So keeping that information straight is going to be important for understanding transfusion reactions and agglutinations, etc. I would go to Blackboard and click on that animation there, and it'll walk you through that as well and kind of animate that for you. Okay, so there's the basis for the ABO blood typing scheme. Now, again, here is just a summary table of this, and so here you have your blood group. If you're AB B, A, or O. Summary of which antigens are on your cell surface. Here's an illustration then of what that looks like. And then the plasma antibodies, right, that are in your plasma. And if you remember from Bio 1 here, this blood type is a codominant situation where you have 3 alleles. And this is just a quick review of some Bio 1. You have 3 alleles. You have a big I A allele, you have a big I B allele, and then there's a little I allele. So we say that big IA and big IB are codominant to each other. Again, this is just an aside. Right? They are codominant, so when you inherit big IA from one parent and big IB from another parent, you're going to be blood type AB. If you inherit big IB and big IB from both parents, then you are blood type B, or if you inherit big IB from one parent and little I from the other parent, you are still going to be blood type B, and so on and so forth here for blood type A. And if you're blood type O, that means that you inherited a little I allele from one parent and a little I allele from another parent. So if you know your blood type, you can then reasonably predict what blood type your parents should have been. Right? And if you know your blood type and you know your partner's blood type, then you should be able to reasonably predict the blood type of any offspring that you have. Okay? Just an interesting little link there to Bio 1. Now, knowing the blood types here for ABO is going to be important to prevent any sort of transfusion reaction. So if we kind of draw a line across here and look at the top half of this diagram, here we have blood type A from a donor, right? And the antibody in that is going to be anti-B, right? And so this is the blood type of the donor, right? When you put that blood into somebody that is also type A, it's not going to be a problem because there are no B antigens on the surface of that red blood cell, right? So these are all blood type A, so you don't get any reaction right here. The antigen and the antibody do not match, and so you get no cross-linking there between antibody and antigen because there's no antigens there for that antibody to bind with. Okay. Now, if you are blood type A, right, of the donor, right, then anti-A antibody in type B blood of the recipient, right? So now, if you are type B but you get type A blood, what's going to happen is that since you are type B, you have anti-A in your plasma. And so then you get this agglutination reaction, and this is when the blood clumps together. And you can see why it clumps because the anti-A antibodies in the B blood that you received is now cross-linking, right, with this blood cell with this red blood cell. Right? And then the same thing is going to be happening on a much larger scale with a whole bunch of other antibodies and red blood cells. And so you get this agglutination reaction, and that causes problems, of course, for oxygen transport as well as blood pressure, right, and several other effects. Okay, so you want to make sure that you have the right blood type coming in there. Another blood type is known as the rhesus factor or the RH factor, and this is a rhesus monkey, and this is the rhesus or RH factor because it was worked out in rhesus monkeys here. And so this is whether or not you are positive or negative for this RH factor. And this RH factor is just another surface antigen, right, which is seen here. So what rhesus antigen is present on the surface of the red blood cell? It's D, right? And you can also think of it just as being RH, right? And so they have this D or this RH. So you could be, if you were, for example, B positive, right? That means that your red blood cells would have B antigens on them as well as the RH antigens. And you could illustrate it like this as well, B with then the D, where the B is for the ABO blood grouping, and then RH is that surface antigen, or it's also sometimes initialed or shorthanded D. So if you were then B negative, you would have B antigens on your cell surface, but no D or no RH. And again, you're going to be looking like that. So to practice that, I would go through and say, okay, what does A positive, what does A negative look like? What's AB negative, AB positive, O negative, etc. So illustrating that as a way to practice. Okay. If you know your blood type, you can see if you where you fit into the North American population. Here, find your blood type, and then are you extremely popular, are you extremely rare, right, etc. Blood typing, you'll do this in class, and as a little example, they're going to give you some unknowns, some patients, and ask you to figure out what blood type they are. And you do that by taking the blood and adding a serum here, right, from the serum, which is the plasma. So what's in the plasma are the antibodies, not the antigens. Right? So you're going to add antibodies to the different blood types. Right? So if the simulated blood that you're going to use is type AB, you would expect then when you add anti-A antibodies that there to be clumping, right? That there would be an agglutination reaction. And when you add anti-B to this, you would expect clumping or agglutination reactions there. And this is what that's going to look like. Right? So if you are blood type B, right, this would be your reaction. So this is no agglutination, and this is yes, positive for agglutination. And you should be able to figure out why this is the negative, why there's no agglutination reaction there, and why there is an agglutination reaction there. And then the same thing for A, and the same thing for O. Right? So this is positive, and this is negative for agglutination. And then this is no agglutination, and then this is no agglutination. This is just what they look like. So in class, little exercise we have you do, you'll be able to take some individuals, simulated individuals, not your own blood, right, and test it and come to some sort of conclusion as to what the blood type is. Now in the simulation, they will also have you'll also have anti-RH there, so you'll be able to figure out whether or not it's positive or negative as well in the simulation in class. Okay. So as a way to summarize and practice all of that, you can go ahead and fill in this table. So if you're blood group AB and then positive or negative here, what antigens would you have on your cell surface? What might your genotype be? What antibodies would you have in your plasma? And then as a way to sort of test yourself, which blood could you receive if you were blood type AB, if you were blood type B, right? And why? So this is asking you to understand that whole agglutination reaction and the combining and the mixing there. So if you can figure out this, this column right here, then you'll be able to figure out which one is the universal donor and which one is the universal recipient. Okay? So you should be able to now here describe and identify what an antigen and antibody is. Talk about the ABO blood grouping, identify them, and describe why, what makes you blood type A, what makes you blood type B, etc. What makes you positive or negative, and for that RH factor. And then also be able to explain what an agglutination reaction is. And then in the exercise in class, you'll be able to work on this a little bit more, explaining blood typing, transfusions, the universal donor and recipient. And then also then in class, we'll talk more about this hemolytic disease of the newborn, erythroblastosis fetalis. Okay, the slides that follow this are about that erythroblastosis fetalis, but we can talk about that in class a little bit more. So thanks for listening. If you have any questions, please bring them to class.